Hello, my name is Jessica Mitro, and I'm a junior here at WVU. And my project is on how nod-like receptor X-1 is a novel mediator of ozone-induced lung inflammation. According to the World Health Organization, over 4 million deaths per year are attributed to outdoor environmental pollution. Ozone is one of the most toxic and reactive gaseous components of air pollution. Acute ozone exposure has been found to aggravate chronic lung diseases such as cystic fibrosis and asthma in susceptible populations. So my protein of interest is NLRX1, belonging to the nod-like receptor family. NLRX1 has been implicated in multiple pathophysiological processes, such as oxidative damage, proliferation, mitochondrial dysfunction, and apoptosis. NLRX1, unlike other members of the nod-like receptor family, has actually been found to inhibit major pro-inflammatory signaling pathways, such as type 1 interferon and toll-like receptor-mediated NF-kappa B. Till date, NLRX1 has been studied in diseases such as cancer and diabetes, but its role in the modulation of innate immune responses due to environmental pollutants is not yet clear. We hypothesized that NLRX1 plays a significant role in inhibiting ozone-induced lung inflammation. So for this project, we used C57 black 6 mice as wild type and NLRX1 knockout mice. The mice were randomly divided and exposed to filtered air as the control, or 1 ppm of ozone for 3 hours, followed by euthanization 24 hours post-exposure. The bronchoalveolar lavage, which is a sterile PBS wash of the lungs for collection of inflammatory cells, was performed. Lung tissues were also collected and frozen for further experiments. Now, Figure 5 contains the bronchoalveolar lavage fluid. Data. In Figures A and B, you can see that the total cell and macrophage counts are significantly higher in NLRX1 mice, knockout mice, compared to the C57 wild-type mice post-ozone exposure. Additionally, in Figure C, we found a significant increase in neutrophils in mice lacking NLRX1 compared to the C57 wild-type mice post-ozone exposure. This states that there is a much higher level of inflammation in NLRX1 knockout mice as compared to the wild-type animals. Now, Figure 6 shows the differential cells in the bronchoalveolar fluid. In Figure D, which represents the NLRX1 knockout mice, there are significantly higher numbers of neutrophils compared to the C57 wild-type mice, which is represented by Figure B. Figures A and C are control samples without any neutrophils. Next, we performed real-time PCR in the lung tissue to identify the fold changes in inflammatory genes. Now, Figure A represents KC, which is a neutrophil chemoattractant and is expressed significantly higher in NLRX1 as compared to the C57 wild-type mice after ozone exposure. We also found significant increases in different pro-inflammatory genes like IL-6, TSLP, IL-1B, IL-13, and TNF in NLRX1 knockout mice compared to the wild-type mice. Now, Figure 8 represents the gels that I ran to identify the wild-type mice and NLRX1 mice, both of which we breed at WVU. Therefore, in conclusion, we have found that ozone exposure causes significant increase in neutrophilic infiltration in NLRX1 knockout mice compared to the wild-type mice. We also found that NLRX1 knockout mice demonstrate significantly higher gene expression of pro-inflammatory cytokines compared with wild-type animals following ozone exposure. In the future, we plan to quantify the inflammatory proteins after ozone exposure in both NLRX1 knockout mice and wild-type mice via techniques like Western blot and ELISA. Further, we would like to investigate the role of NLRX1 in the modification of the NF-kappa B signaling pathway in ozone-induced inflammation, as well as assess the role of ozone inhalation on cells of the mediastinal lymph nodes in NLRX1 knockout and wild-type mice. We would also like to study the mechanistic role of NLRX1 in mitochondrial damage induced by ozone exposure. I would like to thank Dr. Hussain for his guidance, the WVU Inhalation Facility, Travis Goldsmith, and Kevin Engels for their excellent technical assistance.